Good morning, class. Welcome to Icon Education. Today we are going to practice Chapter 9 Biomolecules Exemplar Questions. Question number 1. It is said that elemental composition of living organism and that of inanimate objects like earth crust are similar in the sense that all the major elements are present in both. Then what could be the difference between these two groups? So students, you need to find out what is the difference if all the elements present in the living body and earth crust are same, then what makes the difference? So if you look at the option 1, living organisms have more gold in them than inanimate object. It is incorrect because gold is not there. Living organisms have more water in their body than inanimate object. This is also incorrect. Okay. Living organisms have more carbon, oxygen and hydrogen per unit mass than the inanimate object. This option C makes the difference. Okay. Why? Because we know that in the table it is given carbon. Carbon in the earth crust is 0.03%. And in the human body it is 18.5%. Similarly, hydrogen in the earth crust is 0.14% and in the human body it is 0.5%. Oxygen in the earth crust is 46.6 and in human body it is 65.0. But student, you see the combination carbon, hydrogen and oxygen, the combination what they are making is protein, carbohydrates, nucleic acid, lipids. They are making biomacromolecules, okay, which is more per unit mass than in the inanimate object. So option C is correct. Okay. Question number 2. Many elements are found in living organism either free or in the form of compounds. One of the following is not found in the living organism. So not found in the living organism is option A, silicon, because in the living body the silicon is almost negligible. Okay. Question number 3. Question number 3. Amino acid, as the name suggests, have both an amino group and a carboxylic group in their structure. In addition, all natural occurring amino acid, those which are found in protein, are L amino acid. For this, you can guess from which compound can the simplest amino acid be made. See students, when you look at the question, here it is asking you L amino acid. What is this L amino acid? L amino acid. L amino acids are the biologically active left-handed isomers of amino acid that serve as a fundamental building blocks for protein in all living organisms. That means, see the amino acid structure, the alpha carbon is there in the center and it is having a carboxylic group and amino group, a R variable and hydrogen. Okay. So from which of them is the most simplest amino acid? The most simplest amino acid is glycine. In the structure of glycine, we have one amino group, one carboxylic group, one hydrogen, and the R variable in glycine is hydrogen, H. R is H in case of glycine. Okay, students? Next. Question number 4. Many organic substances are negatively charged, example acetic acid, while others are positively charged, example ammonium ion. An amino acid under certain conditions could have both positive and negative charges simultaneously in the same molecule. Such a form of amino acid is called. We know that amino acid is having a ionizable nature. Okay, the amino group and the carboxylic group can show ionizable nature in the solution. So, alpha carbon, R variable, hydrogen, and NH3 positive and COO negative will form a Zwitter ion. Zwitter ion is a dipolar ion. It is a neutral ion. We are having both the positive and the negative ions. Okay. So such a amino acid is called as Zwitter ion. Question number 5. Sugars are technically called carbohydrates, referring to the fact that their formula are only multiple of CH2O. Hexoses therefore have 6 carbons, 12 hydrogens and 6 oxygen atoms. Glucose is a hexose. Choose from among the following that is another hexose. So a hexose sugar will have C6H12O6. So glucose is a hexose sugar. Similarly, the isomer of glucose is fructose. Fructose molecular formula is also C6H12O6. So fructose is also a hexose sugar. Now look at the other option, erythrose. Erythrose is C4H8 and O4. It is a 4 carbon carbohydrate. If you look at the ribulose and ribose, they are C5H10O5. Both are pentose. Okay. So the correct answer is fructose is a hexose sugar. Question number 6. When you take cells or tissue pieces and grind them with an acid in a mortar and pestle, all the small biomolecules dissolve in the acid. Protein, polysaccharide, and nucleic acids are insoluble in mineral acid and get precipitated. The acid-soluble compounds include amino acid, nucleoside, small sugar, etc. When one adds a phosphate group to a nucleoside, one gets another acid-soluble biomolecule called. See, here they are giving a lengthy question just to consume your time. Here the main line here is phosphate group is added to a nucleoside. So nucleoside is formed by nitrogen base plus sugar. Okay, that is pentose sugar will give rise to nucleoside. If phosphate is added to it, then it will become a nucleotide. So the correct option is option D, nucleotide. Question number 7. When we homogenize any tissue in an acid, the acid soluble pool represent. This is a direct question. The acid soluble pool represent the content present in the cytoplasm. So the correct option is option A, cytoplasm. Question number 8. The most abundant chemical in living organism could be. The most abundant chemical is water. Okay, water is 70 to 90%. 70 to 90%. Okay, cellular mass 70 to 90% is there in the living cell. Question number 9. A homopolymer has only one type of building block called monomer repeated n number of times. A heteropolymer has more than one type of monomer. Proteins are heteropolymers of amino acid, while a nucleic acid like DNA or RNA is made up of only 4 types of nucleotide monomers. Proteins are made of. We know that amino acid are the building blocks of protein. That is, proteins are made up of a monomer, that is amino acid, which are 20 different types of amino acids are there, which is making the structure of protein. All are amino acids. That's why we call them monomer. Why it is called a heteropolymer? Because these amino acids are different, like glycine, valine, serine, etc. Question number 10. Protein perform many physiological functions. For example, some functions as enzyme. One of the following represent an additional function that some protein discharge. Okay, so these are the functions of protein. Protein act as a antibiotics. Protein act for they make pigment conferring the color to the skin, pigments making colors of the flower. Additional function performed by the protein is some proteins can also be hormones, like when you talk about insulin. Okay, so the correct answer is hormones. Question number 11. Glycogen is a homopolymer made of. This is again a direct question. Starch, cellulose, glycogen, they are the homopolymer of glucose. They are made up of glucose units. Question number 12. The number of ends in a glycogen molecule could be. The number of ends in a glycogen molecule could be. We know that the glycogen molecule is having the branched structure. Okay, they are the branched polysaccharide. For example, this is the main chain which is having 3 branches, and here 3 branches. So each branch will have one non-reducing end. So 1, 2, 3, 4, 5, 6. 6 non-reducing end, and the main branch will have one non-reducing end. So the answer is equal to the number of branches plus 1. The number of ends in the glycogen molecule could be equal to the number of branches plus 1. So 1, 2, 3, 4, 5, 6 ends plus 1 end of the main chain. So the correct option is option A, equal to the number of branches plus 1. Question number 13. A pure protein should normally have. A pure protein should normally have how many ends? We know that the protein will be having 2 ends, that is called as N-terminal and C-terminal. The N-terminal represent the first amino acid which is present at the left end, and the C-terminal represent the last amino acid which represent the right end. So a pure protein should normally have 2 ends. Question number 14. Enzymes are biocatalysts. They catalyze biochemical reactions. In general, they reduce activation energy of reactions. Many physicochemical processes are enzyme mediated. Some examples of enzyme mediated reactions are given below. Tick the wrong entry. So students, what you need to find here is which of the following reactions will not need any enzyme. If you look at the first option, dissolving carbon dioxide in water does not need any enzyme because it is happening by the process of diffusion, where it is a physical process which don't require any enzyme. If you look at the second option, untwinning the 2 strands of DNA, it needs an enzyme, DNA helicase. DNA helicase is the enzyme which helps in unwinding. Look at the third option, hydrolysis of sucrose. Sucrose will break down into glucose and fructose, which needs an enzyme called invertase. So it is also an enzyme mediated process. The fourth option, formation of peptide bond, it needs an enzyme, peptidyl transferase. Peptidyl transferase. So that means the rest of the 3 needs an enzyme, whereas dissolving of carbon dioxide in water does not need any enzyme. So with this, we have finished the practice questions of chapter 9, Biomolecules. Hope you find it easy. Thank you.